Hi friends, this is Preeti. Today I am going to discuss about insulin. Introduction. Insulin is an anabolic peptide hormone produced by beta cells of pancreas called as islets of Langerhans. So pancreas can produce alpha cells and beta cells. So from the beta cells, this insulin is produced. Insulin helps in the regulation of blood glucose level. So if there is a high blood glucose level, it means it is a diabetic condition. Diabetes is of two types, type 1 and type 2. Type 1, people with type 1 diabetes don't produce insulin. So the blood glucose level is increased in type 1 people because they don't produce insulin. In type 2, people with type 2 diabetes don't respond to insulin. Later in the disease, they don't make enough insulin. So in this type, the insulin is produced but they don't respond to the produced insulin, and in severe conditions, as this situation is prolonged, they don't make enough insulin. And the next one, human insulin protein is composed of 51 amino acids. So this is the structure of human insulin. It consists of A chain and B chain. A chain consists of 21 amino acids and B chain consists of 30 amino acids. A chain and B chain are linked together by two sulfide bonds, and there is an additional disulfide bond is formed within the A chain. So here we can see the additional disulfide bond within the A chain, and these are the two sulfide bonds between the A chain and B chain. Bacteria used for the production of insulin is E. coli. The INS gene present on the chromosome provides the instructions for the production of hormone insulin. So the INS gene, which is present on our chromosome, is responsible for the production of hormone insulin. Applications of rDNA technology and genetic engineering. So how insulin is produced using rDNA technology and genetic engineering. Here we can see this is the bacterial cell from which we isolate the plasmid DNA, and this is the human cell. Here we will isolate the gene which is responsible for the production of the insulin, that is nothing but the INS gene. So human insulin producing genome. This is the plasmid DNA. So here we use DNA is cut with the restriction enzymes. So with the restriction enzymes we will cut both these plasmid DNA and human DNA. Then this bacterial DNA with human gene inserted. It means we have inserted this human insulin gene into this plasmid. So this is the whole bacterial DNA. Now this plasmid is reintroduced into the bacterium. Engineered bacteria multiply producing insulin. Insulin is separated and purified to produce human insulin. So this is the human insulin. Then insulin is injected into the patient. So here we can see pre-proinsulin, which consists of A chain, B chain and C chain. Three chains are present. This is the C terminal end, this is the N terminal end. So removal of the single peptide will get the proinsulin, and from this a disulfide bridge formation. Due to the disulfide bridge formation we will get this structure, and the removal of C chain will get the active insulin. So by removal of the C chain, finally we will get active insulin, which consists of two chains, A chain and B chain. A chain of 21 amino acids and B chain of 30 amino acids. So this is another method for the production of insulin. Here we are taking insulin A chain and insulin B chain, which are inserted into the plasmids containing antibiotic resistance gene. So this is the ampicillin, which is the antibiotic resistance gene. So here we are inserting into the bacteria, transferring to the E. coli. So we are inserting into the E. coli, then culture the cells. We have to increase the number of cells. We are culturing the cells, then purify the beta galactoside insulin fusion proteins. We have to purify those. Then we'll get the A chain and B chain. Purify A and B chains. Then we have to join them. So we'll get the active insulin. So this is another method for the production of insulin. In the previous methods we are taking the insulin producing gene and in this we are taking directly insulin A chain and insulin B chain, and we are multiplying those cells and we are producing the insulin. Thank you.